Next speaker is Richard Wang. He's also from, this is sort of the Brett Pezur, I guess, section of the meeting. So, Richard.
All right. Before I begin, I'd like to thank the organizers for giving me the opportunity to talk a little bit about my research today, and I'd like to thank all of you in advance for being here to listen to it. Today I'm going to talk to you a little bit about my work characterizing genetic variation in recombination rate. And for the rest of my talk, when I talk about recombination rate, I'll be referring specifically to the number of crossovers that occur in a meiotic cell. So this overall recombination rate can evolve rapidly. There's substantial variation that's seen both within and between species. And this variation has a number of evolutionary consequences, including affecting the degree of linkage between genes, the levels of genetic and haplotypic diversity, and it also influences the efficacy of selection. There's also some health-related phenotypes linked to recombination rate, like fertility and the rate of non-disjunction. And given these important consequences of variation in recombination rate, one of the questions that we'd like to ask is, what are the genetic underpinnings of recombination rate variation? So I'd like to acknowledge that there are a number of environmental factors that influence recombination rate, including stress, temperature, nutrition, and age. But there's strong evidence that there's a strong genetic component to variation in recombination rate, especially with the discovery of loci that confer differences in this global recombination rate. Today, what I'm going to talk about is using intermediate phenotypes in the recombination pathway to characterize sources of genetic variation in recombination rate. What do I mean by that? So this kind of textbook cartoon shows the pathway to a crossover. So crossovers, as most of you know, begin as double-strand breaks. There's this proteinaceous structure that holds the homologous chromosomes together during this phase of meiosis called the synaptonemal complex. There's a whole host of DNA repair proteins that come in before this crossover is resolved. And the whole point of showing this is to show you that there are a number of steps that occur on the pathway to a crossover, and that we can use intermediates in this step, either upstream or lateral to the crossover itself, in order to better characterize the genetic variation in this pathway. And one of the techniques that we can use to quantify variation in these recombination phenotypes is immunofluorescent microscopy. And in this technique, fluorescent antibodies stain elements of the recombination pathway to quantify this variation. One of the examples is this DNA repair protein called MLH1, which localizes to crossovers, and we can look at the staining pattern to enumerate the number of crossovers in a single myocyte, which shows up in green here. But today, what I'm going to talk about is the synaptonemal complex. And as I mentioned earlier, the synaptonemal complex is that proteinaceous structure that holds the homologous chromosomes together, which is stained in red here. And it's thought that the synaptonemal complex forms a kind of substrate for which crossovers can happen. And this synaptonemal complex length is strongly correlated with the total number of crossovers. And this technique can be used in the house mouse. And recombination has been extensively studied in the house mouse, and in one example of particularly rapid evolution, as Beth Dumont earlier mentioned, two closely related subspecies, Mus musculus musculus and Mus musculus castaneus, show a large difference in the global recombination rate. And previous work by her has identified multiple QTL that are responsible for variation in this global recombination rate. And I'm going to use this data set to characterize variation in synaptonemal complex length. And this is what I do. I developed an image analysis pipeline which takes these images and transforms them into single pixel wide representations. You can see the original image on the left and the single pixel wide representations on the far right there. And by summing up the number of pixels in that representation, we can get an estimate of the total synaptic complex length in that meiotic cell. And here on the right, I'm showing you the variation in mean synaptic complex length for F2 individuals from that cross. And for comparison, on the left is the variation for F2 individuals in mean MLH1 number, or the number of crossovers in that myocyte. Sorry. You'll notice that there's a nice continuous distribution for both of these traits, and you can also see that the musculus mean is substantially higher than the castaneous mean. As I alluded to earlier, there is a substantial correlation between both of these traits, which I show here. And I use this information to map QTL for synaptic complex length and compare it to QTL that had previously been identified for the total number of crossovers, or the number of MLH1 foci. And one of the striking, this is the first time that QTL for synaptic complex length have been mapped. And one of the striking things is that you see that that QTL of large effect for the number of crossovers, or MLH1 foci, also shows up in the QTL mapping for synaptic complex length. Since I mentioned that these two traits are correlated, we wanted to understand something about the connection, the genetic connection between QTL and how they affect these two traits. So we can imagine some models for how QTL might affect both of these traits. We can imagine that a QTL is pleiotropic and affects both the number of crossovers as well as the synaptic complex length. We can imagine that this QTL affects synaptic complex length, and that in turn affects the total number of crossovers. And we can also imagine a case where the synaptic complex length, or the QTL's effect on synaptic complex length, is totally mediated by its effect on the total number of crossovers. And to distinguish between these different models, I'm going to use QTL mapping itself, because QTL mapping is a form of model selection. And this equation on the left shows that the variation in this phenotype 1, that is why P1 is affected by this QTL. And we have this very simple diagram on the right. We can also imagine that this QTL also affects variation in the phenotype 2. So this is still pretty simple. And we can also try to map the QTL, or we can also try to explain variation in phenotype 1 using phenotype 2 as a covariate. And that's what that third equation shows. And if that's the case, we just draw a little line from the phenotype 2 to phenotype 1. But if the addition of phenotype 2 eliminates phenotype 1, eliminates the connection between the QTL and phenotype 1, that is to say that all of the variation in phenotype 1 can be explained by variation in phenotype 2, then we would disconnect that line from QTL to phenotype 1, and that would mean that the variation in phenotype 1 is, or the QTL's effect on phenotype 1 is wholly mediated through its effect on phenotype 2. We can use this type of logic to build a sort of contingency table where the presence or absence of QTL tells us about the underlying genetic mechanism that determines both of these phenotypes. Okay, so that's what I did. I QTL mapped the number of crossovers with synaptic complex length as a covariate. I did the same thing for synaptic complex length with the total number of crossovers as a covariate. And for the purpose of time, I'll only focus on a few of these. So in this example, there was a QTL on chromosome 7 that affected the total number of crossovers, or variation in the total number of crossovers, regardless of whether or not we used synaptic complex length as a covariate. And there's no similar QTL on chromosome 7 for synaptic complex length. And in this case, we can just draw a simple line like this from chromosome 7, or the QTL on chromosome 7 to affecting the total number of crossovers. On chromosome 3, we found that the QTL appears to affect the variation in the number of crossovers regardless of whether or not we use synaptonemal complex length as a covariate, and that it also affected variation in synaptonemal complex length regardless of whether we use the total number of crossovers as a covariate. And in this case, using our logic as before, we would imagine that this QTL pleiotropically affects both the number of crossovers as well as synaptonemal complex length. Finally, I'll move on to the QTL of large effect on the X chromosome. And this QTL appeared to affect variation in the number of crossovers regardless of whether or not we use synaptonemal complex length as a covariate. But it only appeared to affect variation in synaptonemal complex length when we didn't use the total number of crossovers as a covariate. And this is the interesting case where it appears that the synaptonemal complex length is wholly mediated, or the effect of this QTL on synaptonemal complex length is wholly mediated by its effect on the total number of crossovers. And this is kind of interesting because even though our phenotype only had a single time point, right? So this immunofluorescent microscopy captures a single time point. This analysis suggests that the determination of the number of crossovers happens before the determination of total synaptonemal complex length, or at least the effect of this QTL on that determination is such. So to summarize, synaptonemal complex length is a quantitative trait with genetic basis for its variation. We identified 3 QTL for synaptonemal complex length variation. We identified causative roles that individual QTL play in both of these traits. One of the most interesting cases was that QTL on the X, which appeared to affect synaptonemal complex length by affecting total crossover number. And finally, this last finding suggests that the number of crossovers may be determined before the completion of chromosomal synapsis. And I think that's pretty surprising because in many of the models, the synaptonemal complex is supposed to form this substrate on which crossovers happen. All right, and with that, I'd like to take any questions.
That was very interesting. I'm surprised that there actually is much variation in synaptonemal complex length because you sort of have a constant amount of chromatin in all these hybrids, presumably. So what do you think is responsible for the variation in length itself? Is it gaps in the synapsis, or is it sort of a general compression or elongation of a full-length synapsed pair? Where do you think the variation is actually coming from?
Let's see. I think that from the literature, I think that there's a variation in the chromatin loop sizes. So during synapsis, the synaptonemal complex forms a central axis for the recombinations to happen, but the DNA itself is like forming these loops around that central axis. And I think there's some evidence that loop size might be the reason there's variation in the overall central axis.
Thanks.
Yeah, I don't know if you mentioned, I'm guessing that was all in males. Is this SC length different in the parental substrains between males and females?
Yes, I think there's also variation in synaptonemal complex length in females. This data, again, was from males, as you said, but I don't remember the direction off the top of my head.
It's in the same direction? You don't know? In the same direction for the two parental species?
Yeah, I believe so.
Are you not concerned that the differences in collecting these two phenotypes may potentially affect your statistical power differently in the two tests you're doing, and thus when you're like rejecting in the null hypothesis on one end but not on the other phenotype may potentially bias your results?
Sorry, I didn't quite catch that.
The two phenotypes are collected differently, right? So there are different effects when you're collecting these. Right, you're measuring two different things, but you are actually like deducing from not rejecting the null hypothesis to your architecture. So do you think that may possibly bias some of these results, and to what extent may it do so?
So if I understand your question, you're saying that these two phenotypes were collected differently?
Right, there are two different features, right, at the end.
They are two different features.
Yes, so you do have different distributions on them, so it may affect your statistical power, no?
Yes, I think that's the case. It's really hard to hear from
Oh,
back
I'm
there.
sorry, maybe you can
Maybe
take it
we
offline.
can talk about this later.
Yeah.